John, Evelyn, Captain Paul, Barry, Barbara, and Drew, and me. My opening words today are from Neil deGrasse Tyson. Everything we do, every thought we've ever had is produced by the human brain. But exactly how it operates remains one of the biggest unsolved mysteries. And it seems the more we probe the secrets, the more surprises we find. So I have some announcements. We have our next Humanist Happy Hour, the one for March, is going to be on Thursday, March 12th from 6 to 8:30. I don't know, one place I saw 6 to 8:30, one place I saw 6 to 8. Probably doesn't make any difference, right, Greg?
Yeah, the bingo goes to 8:30.
Oh, bingo. Okay, that's going to be at Harry of the Natives. That's a musical bingo night. That's why we're doing it on the Thursday. 11910 Southeast Federal Highway. That's in Hobe Sound. It's a lovely place. It tends to be a little more conservative of a bar, but we don't care. A few more. These are save the date items. We're going to be having a booth at the Sebastian Earth Day Festival. That's going to be on Saturday, April 18th at Riverview Park. We're going to be looking for volunteers to help staff that booth. After the fair, we're going to have a happy humanist happy hour at the Paradolia Bar and Restaurant, which is right around the corner from the festival. 712 Cleveland Street in Sebastian. So that's going to be on April 18th. And that festival is from 9 to 3, which is a very reasonable coverage. So we're looking for people to volunteer, and you can sign up on the website. Also, we have a booth at the Treasure Coast Pride Fest. That's the following Saturday on the 25th. That one lasts a little bit longer. That's from 10 in the morning to 6 in the afternoon.
We are sponsored.
And we are sponsors of that event. And by the way, while I have you here, we have our new Humanist Treasure Coast t-shirts. I have one on, Barry has one on, and Greg has one on. And these t-shirts were specifically ordered for all the folks who signed up and are going to be working at the Treasure Coast Food Bank. So they're going to be a team. So when people go in there, they're going to see the Humanists of the Treasure Coast Food Bank team. And if you are not able to volunteer for any reason, like maybe because you live far away, or maybe you have some constraints on being able to lift 40 pounds, or I don't know what all the restrictions are, but you can order one of these online on our website. This morning, our Humanist Secular Moment is going to be provided by Captain Paul.
A cord that's a little hook down there.
Yeah, hold on, everybody. Curious here. There.
One right there.
Yeah, we have microphone. Oh, you want a microphone?
I have no idea. Do I?
Can everybody hear me okay on Zoom? Yes, okay, thank you. Go ahead.
Okay, so today I'm going to read you. This is actually the introduction to the second edition of the book, All I Really Needed to Know I Learned in Kindergarten. We are sent to school to be civilized, to be introduced to the essential machinery of human society. Early in our lives, we are sent out of the home into the world, to school. We have no choice in this. Society judges it so important that we be educated that we must go. It is the law. And when we get to school, we are taught the fundamentals on which civilization rests. These are first explained in language a small child can understand. For example, it would be no good to tell a 6-year-old that Studies have shown that human society cannot function without an equitable distribution of the resources of the earth. While this statement is profoundly and painfully true, a child cannot comprehend this vocabulary. So a child is told there are 20 children and 5 balls to play with. Likewise, 4 easels, 3 sets of blocks, 2 guinea pigs, and 1 bathroom. To be fair, we must share. Likewise, a 6-year-old will not understand that, by and large, it has been demonstrated that violence is co-productive to the constructive interaction of persons in societies. True, but a child can better understand that the rule out in the world and in the school is the same. Don't hit people, bad things happen. The child must understand this rule is connected to the first rule. People won't share or play fair if you hit them. It's hard to explain the cost and consequences of environmental pollution and destruction to a 6-year-old, but we are paying a desperate price even now because adults do not heed the instructions of kindergarten. Clean up your own mess, put things back where you find them, don't take what's not yours. The history of society is more defined by its understanding of diseases than its formulation of philosophy and political theory. True. Basic sanitation. Keeping excrement off our hands as well as out of our minds is important, but it's enough to teach a child to use a toilet, flush, wash his hands regularly, and so on. From the first day, are we told in words we can handle what has come to be prized as the foundation of community and culture. Though the teacher may call these first lessons simple rules, they are in fact a distillation of all the hard-won, field-tested working standards of the human enterprise. Once we are told about these things, we soon discover we are taking a lab course. We're going to be asked to try and practice these precepts every day. Knowledge is meaningful only if it is reflected in action. The human race has found out the hard way that we are what we do, not just what we think. This is true for kids, for adults, for nations. I'm sometimes amazed at what we did not fully grasp in kindergarten. In the years I was a parish minister, I was always taken aback when someone came to me and said, I've just come from the doctor and he told me I have only a limited time to live. I was tempted to shout, what? You didn't know? You had to pay a doctor to tell you at your age? Where were you the week in kindergarten when you got the little cup with the cotton and water and seed? Life happened, remember? A plant grew up and the roots grew down. A miracle. And then a few days later, the plant was dead. Dead. Life is short. Were you asleep that week or homesick or what? I never said all that, but I thought it, and it's true. The idea was for us to have the whole picture right from the beginning. Life and death, life, death, one event, one short event. Don't forget. There's another thing not everyone figures out right away. It's almost impossible to go through life all alone. We need to find our support group, family, friends, companion, therapy gatherings, team, church, or whatever. The kindergarten admonition applies as long as we live. When you go out into the world, hold hands, stick together. It's dangerous out there, lonely too. Everyone needs someone. Some assembly is always required. What we learn in kindergarten comes up again and again in our lives as long as we live, in far more complex polysyllabic forms, to be sure, in lectures, encyclopedias, Bibles, company rules, law, sermons, and handbooks. Life will examine us continually to see if we have understood and have practiced what we were taught that first year of school. Across the course of our lives, we will wrestle with questions of right and wrong, good and bad, truth and lies, again and again and yet again. We will come around to that place where we came in, to that room where the elemental notions about humanity were handed to us with great care when we were very young. Of course, it wasn't literally all you really need to know, certainly not. But if you didn't get this basic stuff to begin with, you and society will pay a heavy price for your failure. If you did learn it and do practice it, then all the rest of what happened, what you needed to know, has a lasting foundation.
Thank you.
Thank you, Captain Paul. Took us back. All right, I just want to let you know that Al Hall just entered the room. Al was living up in Gainesville. And Al actually attended Doreen's interment. She didn't want to have any flowers or any ceremony, but he was living in Gainesville. But I think now you're in Port St. Lucie, right?
Yes.
You want to share any comments about your experience? We have to pass, I don't mean to put you on the spot, but it's up to you if you don't want to. You already did. Can you pass the mic back to Al? We were just happy to have a member be there.
Okay. Gainesville has a cemetery for people who have been buried naturally. Can't remember the name of it, but
Prairie Creek Conservation
We
Cemetery.
can't hear him very well. Joanne.
Okay, is this better?
Yeah.
So she was interred in Prairie Creek Conservation Cemetery, a cemetery that I happened upon riding a trail one day. I showed up in the back of the cemetery. What they do is they bury without chemicals. In other words, no embalming, no casket. They bury about 3.5 feet deep, wrapped in a shroud. And the reason for that is they want the body to break down and return the minerals and so forth to the environment. If you bury deeper than that, then the body doesn't break down. They have a small cart that has wagon wheels on it that you can hand pull. They put the shrouded body on the cart, asked me if I wanted to pull the cart. So he pulled the cart down the dirt path out into the woods. They helped me push it. We got there, lifted the body on a board, set it above the grave, and then lowered it into the grave. Asked me if I wanted to say a few words. So I mentioned her, dedication to the environment and being a good person. I really didn't have a speech ready. Asked me if I wanted to throw the first handful of dirt on. So I did that. And then we covered her. I'm sure she would have loved the ceremony. Very appropriate for her. I took some pictures. They took some pictures of me pulling the cart. I don't think she would want them to be shared. So we're not.
Thank you, Al. I'm sorry, I didn't mean to put you on the spot, but I think the fact that you were there, now you've allowed us.
I expected to have to say something.
Okay.
It's okay.
Good. All right. All right, everybody. All right, so, and thank you for being there. And now, are you a resident of Port St. Lucie now? Or Stewart?
Yes, I'm up on off Jennings Road.
Okay,
You know where Jennings Road is?
Yeah.
That's where the guy with the cross used to stand.
yes, right.
There's a small RV park back there. And I'll be
Okay.
there for 2 months.
2 months. Okay, great. So we'll see more of you then. All right, everybody, let's get to our program today. We're going to watch a video. This is a TED Talk video. It's entitled Growing Mini Brains to Discover What Makes Us Human. It's a very science-oriented video, and I'm going to share it with you now. That's my plan anyway. All right, can everybody see that? Can I get a thumbs up from somebody? All right, good. Let me start it. Where's my mouse?
I was gonna do some intro.
Oh, I'm sorry.
No,
Before we start this, Captain Paul's going to do an intro.
Well, my idea what I was going to do for an intro changed yesterday, actually. First off, I just want to say that
I'm going to move this
several of us
over.
here are on the program committee.
I'm sorry, could he speak a little louder?
We're going to, I have to move my laptop over
Yeah,
again.
yeah, it's all right. We didn't have the mic
No, no, no.
in appropriate position here.
Great.
Anyway, several of us here are on the program committee, and we each bring a different perspective to it. we each bring a different perspective to it. Paul Ivers, who usually only attends this meeting and the program committee meetings, has a particular function in my mind. He's the canary in the coal mine. He's the one, when I bring up an idea, he's the one who always says, what has this got to do with humanism? And so it kind of keeps us on track a little bit anyway. So my rationale for this video about organoids was several. One, I've always been fascinated by self-organizing systems, which is what part of what this is about. Life itself may be a self-organizing system. When I saw my grandson grow from 3 pounds to 5 pounds while living in a glass box in a NICU, it gave me an appreciation for self-organization. I mean, his mother wasn't doing anything to grow him anymore. He was just in a glass box in a hospital. The other thing, but what happened yesterday was this article came in. I just thought I would read a little bit of it just because it was a better introduction to this video probably. And we'll get to what it has, why I thought this pertained to humanism. In 1907, American biologist Henry Van Peters of Wilson discovered that sponges will reform into living creatures after their cells are broken apart through a fine mesh screen, demonstrating that the cells of living organisms contain something telling them how to build complex multicellular structures. Researchers dined out on this for decades. Their subsequent discoveries led to the isolation of pluripotent stem cells, master cells, that can divide endlessly and become any type of cell in the body, first from mouse embryos in 1981, then from human embryos in 1998. Lab-grown brains finally came onto the scene in 1913 when a team of scientists led by Madeline Lancaster created the first brain organoid, tiny three-dimensional cell creature mimicking the human brain. These mini brains made out of stem cells contain real neurons, thus allowing researchers to study brain development, model neurological diseases, and test drugs from human trials. As you might guess, the practice is not without controversy. And the news was now scientists at University of California, Santa Cruz are taking lab-grown brains into their toddler era, demonstrating that brain organoids can process information in real time. So you'll see in the video, she's interested in why humans generate more neurons than other animals. Other people have moved on to using organoids to do simple computations. 2021, a group taught some organoids to play Pong, and this group has taught organoids how to mimic the action you do when you hold a broom upright on the palm of your hand by its handle, which is a long-standing test for humanoid robots. Can they balance a broom standing? So anyway, so those two things. What was interesting is that not only do we know why humans develop more neurons, but we're now seeing organoids do human-like computations. And then the question would be, will they become conscious? So I think we are moving over into why humanists would be concerned about that development. It's like a parallel path to AI in a way. I mean, growing, just as a factoid, the new plans for data centers take about a gigawatt of power. Human brain takes 20 watts. Let's go to the video.
Okay, thank you.
Thank you.
All right, let's play. As I was talking, I knocked over my water. Never a dull moment. All right, so with that intro, let's take a look at this video. Thank you, Greg.
It's too tiny.
I'd like to start
with a very simple question. It's perhaps the oldest question ever to be asked, and it's simply, why? Why are we here? Why are we human? Why are we so special in the animal kingdom? And why can I ask that question and no other animal can? I mean, we humans are deeply curious. As children, right from the start, we begin exploring our surroundings, experimenting with the world around us, and seeking out answers to questions we didn't even know existed. Haven't you ever noticed how children will ask never-ending whys that can drive you nuts? I basically never outgrew that why phase of childhood. I'm constantly wondering why, like, I don't know, why do I have 2 eyes and not 8 like a spider? That would be weird, right? But that big question, why are we human? Why are we special? That's something I always come back to. But, I mean, you all know why. It's your brain. Okay, so what makes our brain so special? I mean, think about it. Right now, your brain is thinking about what makes it capable of thinking, about what makes it capable of thinking, about, okay, I'll stop. You get the idea. So basically, my brain is trying to figure itself out. It's too bad I can't just ask it why it's so special. So I'll have to find another way. So for millennia, scientists have been dissecting and examining brains of a wide variety of different animals, including humans. And one thing is clear, the human brain is big. But it's not just that it's big, because there are other animals with larger brains, like the elephant brain, which weighs about 5 kilos. It's about 3 times bigger than the human brain. And yet most would argue that the human brain outweighs the elephants in terms of intelligence. So what makes a given human, like this one, so smart? Well, it turns out that it's not just size, but actually number of neurons. So the human brain actually has more neurons than an elephant in the cerebral cortex, the part of the brain that's responsible for higher thinking like logic and reasoning. So to give you a sense of the scale that I'm talking about, I'd like to start by looking at a galaxy. So this is one of about 100 billion galaxies in the known universe. That's a lot of galaxies, right? But believe it or not, that is the same number of neurons in your brain right now. And what's more, in the largest galaxies, there are about 1 trillion stars. But that is only 100th of the number of neural connections in your brain. As I'm speaking, your brain is lighting up with neural activity at points of contact called synapses. This is how your brain cells communicate with one another, and it's what's responsible for your brain's enormous computing power. In this room alone, our brains combined have 60 quadrillion synapses. That's 60,000 times the number of stars in the largest galaxies, and 600,000 times the number of galaxies in the entire universe. Okay, so it's clear, to understand what makes our brain special, we need to understand why it has so many neurons. Where do those neurons come from, and what determines their numbers? So for obvious reasons, we can't very well start probing a developing human brain. So early in my career, I took a pretty much standard approach of looking at brain development in rodents. And animal models have given us a number of really fundamental discoveries. Like, for example, we now know that deep inside the developing mammalian brain, including humans, neurons are generated from a special stem cell type called a neural stem cell. And scientists have been probing these neural stem cells to try to figure out what makes them tick. But the question is, why do a human's neural stem cells generate so many more neurons than those of, say, a mouse? And that's something that's always nagged at me. But how can you study one of the most fundamental questions, what makes our brain unique, if you can't touch it? It's like, imagine asking a child to try to figure out how to fold out solar panels on their toy Juno spacecraft here work, and then telling them that they can't touch it. I mean, their hand would just be twitching, wanting to fiddle with it and turn the crank at the back and see what happens. And that's the problem that neuroscientists have been faced with when it comes to the human brain. Okay, so we can't play with a human brain. But one way to understand the toy spacecraft is to build it. So simple, build a human brain. That might sound crazy, I know, but let's just think about it for a second. So when I started tackling this problem, there were a number of really really important technologies that had just come out. And so the first one I want to talk about are stem cells. Stem cells are really cool. Now I know that the patchy cells at the bottom of this dish don't really look like much, but they can actually become any cell in your body. Scientists can direct them to become a wide variety of different tissue types, including neurons, simply by providing the right combination of different factors. And so we thought, well, maybe, just maybe, we could build our brain by starting with stem cells. But our brain isn't 2D like the cells in this dish or like many other methods for generating neurons. And so we thought, well, maybe we need to go 3D. So I spent many hours in the lab culturing stem cells in little 3D balls like the ones you see in this dish. And just like trying to build a toy spacecraft with no instructions, I had to try a lot of different things, lots of different factors and nutrients like sugars and vitamins and timing. And there were a number of previous studies that identified important components in supporting brain cells. And so we tried those in combination with 3D, and the results were promising, but the real game changer came in a special protein gel that we embedded the tissues into. Now, this protein gel causes brain tissue to form with the same structure as it does in a developing embryo. It kind of acts like the supportive tissue surrounding the developing embryo. And when you combine that with what's called a spinning bioreactor, which actually stirs the fluid and agitates the tissues, they could grow to be several millimeters in size. And so finally, we had developing human brain tissue. Right? Isn't that beautiful? So I know that many of you are probably not that familiar with what developing brain tissue should look like, so I'll try to explain. So this is what we call a brain organoid. It's an organoid because it's not like the organ, but not quite. I'll show you. So here's a diagram of the developing human brain, and here's our organoid. And if you were to look inside a developing human brain, what you'd see are these large fluid-filled spaces called ventricles. And there they are in our organoid as well. And lining those ventricles are the neural stem cells, just like in the actual developing human brain. And if we take our organoids and we cut them up into slices and we stain them for different brain cell types, we can actually recognize different brain regions. Like, for example, at the top of this one, you can actually see a large cerebral cortex with a big fluid-filled space. That's the ventricle. And in purple, you can see the neural stem cells lining that ventricle. And on this organoid, you can actually recognize the retina, that brown tissue there, which is actually the neural part of the eye. But in our organoids, these different brain regions are a bit jumbled. Kind of like how a toddler might try to build a toy spacecraft with the solar panels backwards and the detection equipment upside down. And that's because our brain organoids don't have the surrounding fetal tissue to help guide them. And so just like the jumbled toddler-built toy spacecraft doesn't really work like the toy that it should, our brain organoids don't really think like a brain. But that doesn't mean that we can't learn from them. Because if we focus on specific components, like, for example, the cerebral cortex, which I'm showing in a close-up here, you can see that it develops with the same organization as the brain in an actual embryo. You can see neural stem cells in red and neurons in green, and you can see that they're identical in their organization. And that's really remarkable, I think, because basically the way to build a brain in a dish is simply to direct stem cells to become neural and then allow them to do what they're programmed to do, to build a brain. Simply by providing the right conditions to support and nurture the cells in a 3D configuration, the brain is able to self-organize and build itself. And that's really what it does in an embryo, if you think about it. And so now we have this remarkable 3D developing brain tissue that generates these really beautiful neurons, just like neurons in an actual brain. We finally have developing brain tissue that we can actually touch. We finally have something we can play with. And so what can we do with it? Well, one idea is to use it to model neurological diseases. Now, for many reasons, animals don't always make great models of neurological diseases like autism and schizophrenia. As you might imagine, it's a bit difficult to ask a mouse if it's hearing voices or to test its language abilities. And so what we need is a human system. And that's where we think our organoids come in. A good example is the disorder microcephaly. Now, patients with this disorder have smaller brains, and it's not totally clear why, because when we try to study it in mice, they don't show much of an effect on brain size. And so we decided to take our organoid approach. So we took skin cells from this patient with microcephaly, shown here on this MRI, and we generated stem cells, and then we generated organoids. And amazingly, the organoids were smaller and had fewer neurons. So now we have this tool to actually study this disorder, try to figure out what might be the cause. And it turns out that there was a problem with the stem cells, with the neural stem cells, that they were generating neurons too early and becoming depleted so that there weren't any left to generate new neurons. And so this gives us new insight into this disorder, but it also gives us insight into what's required for human neural stem cells to generate neurons and at what point. And so that brings me back to my big question: what determines how many neurons human neural stem cells make, and why do they generate so many? And so to get at that, we look at genes that we think might be important for neuron generation, and we start mutating them. And we use another really exciting new technology for this called CRISPR. CRISPR is a method for introducing specific mutations in the genetic code. It uses something called Cas9 that cuts the DNA in a specific place, like, for example, a gene important for neuron generation. And then the DNA goes back together with a mutation. So in this way, we can introduce mutations that specifically disrupt genes, destroying them or introducing more mild mutations, and we can see what happens to the organoid. And so we're doing this for a number of candidate genes that we think might be important in human brain evolution. But not only that, because we're doing all of this in a dish, we can even make organoids from other animals. All we need are a few skin cells from a human and, say, a gorilla, and we can make stem cells and then organoids and compare them. And not only that, we can also watch cells in real time by labeling them, like the green neural stem cells here, and we can watch them as they generate neurons in the organoid and ask why they generate so many more neurons and what regulates that in humans. And so now we have this really amazing new tool. Brain organoids provide a really remarkable tool to study everything from neurological disease to human brain evolution, and hopefully one day for drug discovery and toxicity testing. But it's also a remarkable system because of its amazing ability to build itself, which is really what it does in an embryo. So now we have developing human brain tissue that we can actually touch. We can take it apart, we can build it, we can play with all of its different components. It makes me feel like a kid again, but with a grown-up question.
Okay, kind of a different kind of topic, very scientific focused, but I'm hoping that it has generated some questions and comments. So can we get the lights back on? Oh, Barry's going to get it. Thanks, Barry. Does anybody have any comments or thoughts about this video? What its implications might be? We're going to start with Drew.
I found it very fascinating, informative. Didn't know anything about it. A little scary, but if anybody can explain what and how you get stem cells, I'd appreciate it. So.
Well, they're embryos, a lot of controversy there has been about embryos being used to harvest stem cells, but I don't know a lot about it. Captain Paul.
Well, as to the stem cells, what they're doing in this research, they're taking basically skin samples, very small samples of skin, and they treat it chemically and they can turn it into neural stem cells. They can turn into other kinds of stem cells also, but these are the ones in this topic. And so this is at least the ethical dilemma of, you harvesting them from embryos and whatnot, because they're just coming from skin cells. But one of the things that I happened to watch another video and a light bulb went on, I hadn't realized that when you do this, the stem cells you get are that person's stem cells. I mean, they're unique to that person. They have their genetic code. So when she talks about using CRISPR, you can use a person's own genetic code and use CRISPR to change the genetic code of that person's cells. So we're getting to the point of being able to do some genetic manipulations that feed back into real people. And in case anybody was real excited about this topic, 35,000. You're a legitimate laboratory, of course. You can buy organoids all grown. They come in a box like a small shoe box, and they have input-output connections, so you can experiment with them. You can use them to do computations or whatnot. So it's kind of fascinating. I don't know at what point you might be able to grow something that you could start thinking is conscious, but we're a long ways from that, but you can see this, we're in the foothills, I think.
Anybody else want to weigh in on this? Let's go to Greg.
It's got some interesting implications. I mean, for medicine, particularly if you could grow a brain type organoid, and then like he said, you could introduce genetic mutations intentionally and see what the effects are on the brain. So there's a lot of medicinal applications for this stuff. But yeah, like what you just mentioned about using organoids as some kind of like organic computer. I mean, yeah, that's getting into some creepy territory there, because like he said, you could grow them and hook them up together and use it as a computer of some kind, and then at what point, where's the line between organoid and consciousness of some kind? It's creepy as heck.
Could you pass the microphone to John? Did you have your hand up, John? Oh, I'm sorry. Okay. Let's go to Linda. I thought you might be online today, Linda.
Yes, this is a little bit in my wheelhouse. That was an amazing video. I haven't kept up with the sort of organoid technology. I mean, for a long time, scientists have been able to create tissue using these 3D matrices and so on. But this is really super interesting that it can organize itself into an organ or a pre-organ similar to the human brain. So yeah, lots of possibilities. And just so you know, stem cells are, especially adult stem cells, you can buy these from companies. And I have a little stem cell company that's very focused. We get stem cells from a few different types of tissues, and we buy them from a company and we grow them in our lab, and they're not hard to grow. You can only grow them for a few passages because they start to slow down and then they start to sometimes differentiate. Like, for instance, we grow these adipose stem cells, which came from usually liposuction from patients. And so they grow out the stem cells and we grow them. And after a while, they actually start making fat in the tissue culture vessel. And that's when we stop using them, because my application is we're generating these great molecules that stem cells secrete, and we're incorporating them into anti-aging, wound healing products. And some of them are very powerful. And this is not using the cells, but just the molecules that they secrete. It's a SRM, stem cell release molecule technology. And they're very powerful with things like preventing radiation burns from people being treated for cancer. That's one of my favorite applications that some of the products are being used for. But yeah, it's amazing. Stem cells are truly amazing. And I think there'll be a lot of new stuff coming out in the future with all sorts of applications.
Can I ask you a question and then I'll get to Juanita? Can I ask you a question? When they ship you stem cells, what condition are they maintained in? Is this, do you get them in an envelope? Does it come in a refrigerated unit? Is it, does it come in a
good question.
vial? How does, I'm thinking that stem cells are a biological item that needs to be shipped in a protective way, right?
Yeah, yes, you're exactly right.
You can't dry them out and rehydrate them, right?
Right. Yes, they come, they're in a little tiny vial, usually just like 1 cc, and they're quite expensive. We get about 500,000, let's say, of these adipose stem cells. It's about $900, I think. And to do what I do, they come on dry ice in a styrofoam box, and they have to immediately be put into the vapor phase of a liquid nitrogen freezer. So they have to be stored in ultra cold conditions. That's like minus 150 degrees centigrade. And then when you're ready to put them in culture, take that little vial out of the freezer and you just basically plop it in a little float in a warm water bath, and they thaw very quickly. And all this has to be done in a cell culture facility where you have sterile conditions. You know, we're all gowned up with all sorts of personal protective equipment. And the cells themselves, you transfer them from their little vial into a flask where they attach to the plastic. And then you put them in a cell culture incubator. And the incubator has to be really controlled. It's high humidity. It's 37 degrees centigrade, which is our own body temperature. And it gets particular gas pumped into it so that it's mimics what the cells would see inside your body. And if any of those conditions are off, the cells won't behave correctly when they're growing. But we grow from 500,000 to, we grow these in these multi-layer flasks, and we routinely get 100 million cells per flask. And then we just culture them and keep harvesting our golden stem cell release molecule mixture. That's what we do. But we can freeze the cells again. I do that, harvest them off the plastic, lift them up, and then put them in a special media for freezing them. And then I ship them up to University of Florida on dry ice, and they put them in there in a liquid nitrogen freezer. So next time we need to grow them, we can just get it from them and not pay $1,000 from the company. So.
Wow. Okay. Thanks for that. I'm getting a better visual idea of how this all works.
Probably more than you wanted to know, but
no,
yes.
No, that was good. I appreciated it because I'm trying to figure out, I know nothing about this. Juanita.
Well, I would follow up with Linda. What do you do with these? I mean, why are we doing this? And what can you do with it once you've grown them?
Are you talking about the ones I grow or just in general?
Yeah. What do you do? Why are you doing this?
So we're generating the stem cell molecules. So stem cells, when they grow in culture, they secrete molecules that are sort of regenerative to your skin. Our focus is on the skin, so we
Oh, on
mostly
skin.
grow. Yeah,
Okay.
so we harvest these molecules. Some of them are in exosomes, which is a new kind of big area of research. So they're in these little packages. And when you put that in a cosmetic product, it helps regenerate the skin, kind of
What happened to her?
return it to its youthful condition.
And then you do what?
Well, the company that I work for creating these molecules, they incorporate it into products like moisturizers, serums, and things like that they sell for a pretty hefty price, and people use them on their skin.
And that adds growing skin. You can't inject it to make your skin grow
No, it
better.
doesn't. Yeah, it doesn't, really stimulate. I guess it stimulates cells somewhat because some estheticians, which I'm not real familiar with the way our products are used, but some in the esthetician space, they do all kinds of things to get these products down under the skin, like microneedling and stuff I would never allow to be done to my skin. I don't care how many wrinkles I have. I'm not going to let somebody, put some little needle punctures and put this magic juice in there. I'll just deal with myself the way I am. But a lot of people do that for looking younger.
It's a topical product.
It's a topical, yes.
Yeah, I was looking for cells that you could inject in your brain to add to your memory, to rejuvenate your memory.
I want some of those too. Let's go to.
Is that might be the future, or what do you think?
I mean, who knows? I don't know if they could do some kind of transplant. Yeah, it's hard to know. But I would say the sky's the limit. It's early stages for some of this stuff.
Oh.
Gloria, you're next.
Yeah, I have a couple of questions maybe Linda could answer. One is does this fall under the category of biomedical research, and who is funding this?
So I would say yes, it falls into the category of biomedical research, and probably a lot of the funding at the basic research level, which is done at universities and stem cell institutes and things like that, is from the National Institutes of Health, primarily, I would say. And then, as you probably know, there's lots of collaborations with, big pharma who funds basic research, sponsors the research. I mean, the stuff that I do is not really research. It's just using these stem cells that are commercially available to create a product. So we don't have sponsors. We just sell our products and use the profits to, invest in some investigation. We do a little bit of research, but not a whole lot.
Pass the microphone to Barry, please.
Thank you. What's the difference between biomedical and biotech? Linda?
I think probably there's a lot of commonalities between the two. Biomedical, you would think is more towards medical applications, so addressing disease states, looking for cures. A lot of biomedical research is on creating devices that help in the health space. Biotech is maybe a little more generic. So for biotech, maybe you're creating diagnostic tests and different things like that. I think there's a lot of overlap between the two. I'd have to look it up to see what the specific kind of differences are.
Does anybody else? Drew,
Yeah.
I'm waiting for an ethics comment to come up or question to come up because the ethics of all of this is what occurs to me. But at the same time, it's just like with AI, we just don't know what we don't know about this. We don't know where this is going to go. So it's really hard to even anticipate the possibilities, although she did talk about some of them. So then you start talking about the implications, the possible negative ethical implications, and who knows? I'm sorry, Drew, go ahead.
Well, just a couple things. The first thing I would mention that in the beginning of her video, she made these analogies to the brain, the neurons in the brain, as an example of the amount of stars in a galaxy or a group of galaxies. I'm not sure. Whatever it was beyond my computation, except to be amazed at how many neurons that are running around in our brains, making them work. The second thing is, the second thing I was going to say is that between artificial intelligence and what we're doing here with these organoids and the amount of things going on in research, it's amazing. My life will not be able to see all of this, but it is going to develop in fascinating ways that should be just amazing in the next 50 years. And the other thing is, Linda, do you see anywhere in the stem cells being able to, in a lab situation, to create an organ, a human organ?
I would say probably. I don't know how elaborate it could be because the organoids that she was making, they got to a certain level, but then couldn't progress beyond that and differentiate into all the different components of the brain in this case because it didn't have all the other fetal tissue around. As we learn more about what's needed, there may be, I certainly think there would be progress. Do we ever get to the point where we could grow organs to replace, like for transplant? Maybe. I could see that happening in a few. I'm always hesitant to predict how long things will take because it seems like we're on this exponential curve now of things just happening so quickly. It's very hard to keep up.
So ethically, did you want to can you pass it to Evelyn? Ethically, it takes me back to the cloning, which they've been cloning for quite some time now. When that possibility came up, everybody was really concerned about where this would go and what could possibly happen with this ability to clone. And I know they've cloned sheep. And I don't know today what's going on with cloning, but the whole ethical issue raised its ugly head when that happened. And now it's kind of, more passe. You don't even hear anybody talk about it. But this is, that's what this reminds me of. Evelyn.
So I love the talk, and I know nothing about it, but it really was intriguing to me. And the thing that made me most interested was when she compared the elephant brain to the human brain, and bigger is not necessarily better, which is what I thought. And I have always thought about dolphins and elephants and animals like that, and I think, why are we superior? And is it only because we think we're superior, but do we really know that? And I think her answer in essence was quality is better than quantity. So the fact that we have more neurons than elephants, or I assume dolphins, makes us more intelligent. But my question is, where do emotions come from? And the brain, I think, only shares information, but where do emotions come from? So that's my question.
And do you believe that animals have emotions?
Yes, certainly. Yeah. Anybody who's had a dog would say yes.
We'll tell you, pass that back to Greg, please.
I think emotions are hormones that your body produces.
Or
to make you
testosterone.
feel a certain way, or certainly they're entirely chemical emotions. But no, I mean the implications of this brain organoid technology are mostly for research, right? So if they can grow little mini brains, they can conduct scientific experiments on them, and then like that could lead to treatments for brain diseases like Parkinson's or dementia or Alzheimer's, right? So that's the goal, is they're trying to cure those diseases, you know. But
Yep.
I mean the CRISPR technology in general, you know, is cool because it's leading to a new era of medicine that's basically gene therapy, like out of science fiction, right? Where we can now cure genetic diseases by changing your DNA. And they've already shown that they can do it to cure sickle cell anemia. That's like the first one. So that might be the first genetic human disease that gets wiped out entirely through gene therapy using CRISPR, you know, but eventually, you know, hopefully they'll get to the point where they can grow organs, like fully functional human organs in the lab, because they could grow an organ that matches your DNA, and then when they transplanted it into you, there'd be no need for organ rejection medication because it's your DNA. They actually grew a new liver that's your liver, literally, you know. So that's hopefully where they'll get to with this technology eventually.
Captain Paul.
One thing that Madeline Lancaster didn't mention, because it wasn't the topic of her talk, but besides these things, you know, looking so much like human cells, they actually started generate the same electrical impulses. In other words, they start having brain activity. They don't just look like brain cells and stain like them. They actually start firing through the synapses and whatnot. So there are at least 2 companies in the world that have very impressive videos out there, obviously trying to drum up investment. But one of them is making a device that's very reminiscent of a printed circuit board. It's like a rectangle, maybe 2 by 2, and in the center there's an organoid, and around the edge there are connections there are actually probes that mounted inside the organoid. So they are trying to develop, if you will, an organoid chip, if you will. So there's a whole different path. Madeline Lancaster runs a lab in Cambridge, England, that is really dedicated to the medical applications. But there's this other company, one is in Switzerland and one is in South Africa. I'm not sure why, maybe something to do with government regulations and whatnot, that maybe those environments are more hospitable to their research they want to do. But anyway, that part intrigued me more because of the open-endedness of it.
All right, we're going to go to next either Liz or Francis, and then we're going to go to Barbara.
So in answer to your question about cloning, it's about 16% successful at this point. People like Barbara Streisand and Tom Brady have cloned their dogs, but you can't clone their personality. And sometimes things can change from genetic variation. So fur color, and things change, environmental exposure. So in other words, people are trying to keep their beloved animals, but they're not really getting their beloved animal back. And to me, this is like learning, just like what Captain Paul was talking about, what we learn in kindergarten. Things are born and things die, and then we have to move on. So to me, this is hugely impactful as far as humanists are concerned regarding cloning, CRISPR, genetics, organoids. So I'm glad we had today's program.
Barbara, you wanted to say something.
It was too smart for me.
You need the mic.
Okay. A couple of things occurred to me. I knew absolutely nothing about this. I hadn't heard anything about it, and it was totally confusing to me. I still am trying to figure out how those organoids communicate. I don't get it. How do we know how they're solving things and so forth? Is it like a computer? Are they attached to some kind of electrical thing? I have no idea. Can someone enlighten me?
Well, Captain Paul is nodding his head, so you might want to pass the
Yeah, no.
microphone to him. In the meantime, we're going to go to Amanda. Amanda.
Hi, I'm glad we watched this today. Just two quick things. First off, just sort of a random bit of info. Our livers actually can regenerate all on their own. So when people need to have a large amount of their liver removed, if there's a certain amount left, the body can regenerate it like half the mass of it in like a month. It's the only organ that does that kind of on its own, which is pretty fascinating. And so people have spent a lot of effort trying to understand the liver and how it's able to do that in order to look at other organs. This CRISPR technology is fascinating. It is the key to probably being able to take care of what are devastating disease processes, like things like Huntington's disease that wipe a person out at 30 years old. But I think it's a long way coming. There's just a lot of research that's being done into it. And just along the lines of talking about cells and humanism and the ethics behind it, I don't know if anybody read the story or has heard of a woman named Henrietta Lacks. She was a woman whose cells were taken without her permission when she had cervical cancer, like back in the 50s. And multiple tons of research has been done based on her cell line that they used taking from her, and her estate just settled with Novartis, a large settlement for taking her cells from her without permission.
Wow. So there's a whole legal.
There was a large settlement from the company that profited off of this.
Wow. All right. Then we're going to go to Captain Paul, then back to Barbara. Captain Paul.
Two things, actually. You reminded me, my stepson, when he was about, I don't know, 7, 8, I forget when, but basically he or someone else shut his finger in a car door and amputated the end of one of his fingers. Maybe, I forget, he lost up to the first knuckle, basically. And we went to his pediatrician. The pediatrician said, I'm reluctant to mention this to you, but I've read studies where up to around age 8, if this happens, your body will regrow the fingertip. He said, I'm a little nervous because I've never seen it done, but the recommendation is just clean it, keep it clean, and keep it protected, and it'll regrow. Should we give that a try? And we said, sure. And it did. It regrew. If you look really closely, you can see a little bit of difference, but it was, and he was amazed because he said, yeah, I read it in a reputable journal, but it surprised him to see it really work. But to Barbara's question, there are two ways that I heard that people, quote unquote, communicated or trained these cell organoids. One, the people, this was in 2021, so it's probably primitive by today's standards. They were training an organoid to play Pong. And when it made the wrong move, whatever, they basically changed the chemicals in its bath to essentially shock it when you get right down to it. Just kind of like, maybe back to grade school when you used to slap someone's hand or something. They shocked it. We've moved on to actually having little teeny electrodes implanted in these things so you can actually intercept their electrical signals and give them on a little electrical shock, much more localized than just changing their bath. So that's kind of where we're at so far on how you do that, how you communicate with them. And you wanted the microphone back.
Yeah. Well, thank you for trying, but I still don't get how as I understood it.
Hold the mic up to your mouth.
I'm sorry.
Thanks.
These organoids, as I understood it,
can solve problems. And I'm wondering, how do we know that? How do they communicate? They can't speak. You know, is there some kind of electrical thing that I can't imagine how they do it. That's my first thing. The other thing is that I was glad that Henrietta Lacks was mentioned. That was the only thing I've had in my history that even comes close to this. And that was one of the most fascinating books I ever read. The reason I wanted to add to that is because the thing that was amazing about Henrietta Lacks was the fact that her cells didn't die. All cells apparently die. They have a lifespan or whatever. And her cells were the only ones that they've ever come across that just don't die. So these researchers or doctors or whatever were selling her cells all over the world without the family's permission or knowledge, because these cells are so unique that they just don't die. The name of the book that I read is The Immortal Life of Henrietta Lacks, and it is an incredibly fascinating book. But anyway, back to how do these organoids communicate with us? I would really love to know. There's got to be some connection, but I don't know what it is.
I think the microphone got shut off, but could you pass it to Drew and you have to get the blue on again.
Are we on?
Yeah, you're on.
Okay. I don't think the organoids communicate. They're just, I think you would think of them as maybe a plant. I don't think they're that organized or that strong, but they are, what you're seeing is a certain amount of cellular activity that can be changed and manipulated and to grow in a certain way. And also, like Captain Paul said, it's interesting to see that biological material can self-organize, which is something unique that we don't even think about. Anyway, I just wanted to mention that we had a good time at the picnic yesterday, thanks to Greg, and it was a good time.
They had a picnic yesterday at the Bush Wildlife Sanctuary, and I guess the weather was good, and everybody had a good time, right?
Yes, it's a very unique place, very fun place.
Yeah, I should have gone. I chickened out with the weather forecast.
Okay.
Yeah. Well, anyway, interesting topic, and I'm with Barbara. I don't read medical journals. I don't keep up with scientific things, and so I don't understand this a lot when I hear about it. I'm trying to think of mostly ethical implications, but you know, at least we present this topic, and we have an awareness now that this stuff is going on, and it's potentially going to likely continue to go on. So if we hear more about it, we're not in the dark anymore, totally in the dark anymore. All right, so thanks everybody for tuning into that, and thanks for your comments. Does anybody have a humanist sharing moment? Anything you want to let people know about that's going on? No? All right. Next week, totally different topics. Next week, our topic is the Black practice of disbelief. And Dr. Anthony B. Pinn is going to be speaking via video. He is on the board of directors of the American Humanist Association, and we tried to get him to be a guest speaker. He's a little bit out of our budget. Hence we went to the video instead, but I think it's really a fascinating video, and I hope you will tune in for that. He is a Rice University professor, and he discusses the principles, history, and communities of Black non-believers. My closing words are from Carl Sagan. Too much openness, and you accept every notion and a hypothesis, which is tantamount to knowing nothing. Too much skepticism, especially rejection of new ideas before they are adequately tested, and you're not only unpleasantly grumpy, but also closed to the advance of science. A judicious mix is what we need. Carl Sagan. All right, everybody, thank you for being here. on another beautiful Sunday in Florida. And I hope we see you again. Ed, I'm going to ask if you could stay on for a minute because I just want to do a microphone test.
Yeah.
Okay, thank you. All right, everybody, take care. Have a great Sunday. Have a wonderful week. We'll see you on Wednesday night.
Your microphone worked well this time.
Yeah, but I'm not sure what's picking up what, so we want to test it. So what I want to do is turn this off. Turn this off.
It was all pretty good till Barbara, like you said, I think she turned the microphone off, and
Yeah.
I turned my computer right up. Drew
All right,
blasted us.
Greg. Sorry. I don't think the lavaliers are working right now. I think the white mic in the speaker that's by my laptop is what carried the day. And if the lavalier isn't working.
Are the batteries charged? Charged
Yeah,
on it?
yeah, we have a charge case.
It has to be a solid blue light.
Yeah, we have a solid blue light. I just want you to take this back there and try it.
Yeah, hang on, I gotta do something with that one first.
Hold on, Ed.
Okay.
Thank you. We won't be long.
No problem. I eat breakfast.
Hey, you want to take this to the back of the room and talk into it? We're doing a test.
It's a job for you.
See, as soon as you get here, we put you to work, Al.
Okay. In the words of the Smothers Brothers,
Let me know if you can hear Al.
I can, yeah.
tasting, 1, 2, 3.
Loud and clear?
tasting.
Loud and clear.
Okay.
Could be a little bit louder, but it's certainly acceptable. I can hear him fine.
Okay, that's all I wanted to do
And
because I
this is
wasn't
just
sure
a
if it was
normal
working
voice,
or
not
not.
loud, not trying to make any.
Yeah, Al came through real well when he first
Okay.
talked about Doreen. So
You can hear him really well. I can't hear him really well. So we have to use both. We have to, because he's at the back of the room, I can't hear him unless he has this. Because the lavalier is good for, it's hooked into my laptop for Zoom, but it doesn't generate sound into the room.
So your speaker on your computer is not projecting like Al's voice?
No. No,
Why not?
because he's not sitting in front of my computer.
No, I'm talking about listening, the speaker, not the microphone, the speaker on your computer. Like where's my sound coming from right now?
My speaker from my computer. Right.
Okay. And
but when when he talks in the back of the room, this lavalier is not coming through on my speaker.
Oh, well that's a problem.
It's going to the people on Zoom loud and clear.
Is there not a separate speaker for it that like the microphone?
What's the same as when you use the mic on the computer, it doesn't play back to you what you're saying.
Yeah, okay.
In a telephone, it would be called side tone. When you talk to the old telephone, you could hear your own voice in the telephone. That's called side tone.
Oh.
All right. So
But there's a problem if it's a mic near a speaker with feedback.
right. So we need to have both still until somebody comes up with something else. All right. Thank you, Ed. I appreciate it.
Sure, no problem. What is plugged into your computer that the microphones feed into?
I have this little thing that goes into my
A magic box of some kind. Yeah.
Yeah, this little thing goes into one of my outlets in my laptop.
Well, I wonder if there's some setting on your computer that would allow the speaker or the audio from that to go through the speakers on your computer. Because we're certainly getting them out here on Zoom. It's coming through loud and clear.
So you're saying a setting on my laptop that
Yeah,
would,
if the device, the dongle, whatever it is, plugged into your computer, maybe in the sound settings, um, there's some way to play back through the speakers. I don't know. I'm shooting in the dark here. I have completely lost your audio at this point, if you can hear me. No audio, can't hear.
can you hear me now?
Yes, that did it.
So when you take this thing out, I put this back in and then you couldn't hear me and I took it out and then you could hear me because when I have this
You
in,
shut all the mics off. That's why he couldn't hear you. And then so when you pull the receiver out, it switched
I
back
know
to
when
the
I
laptop.
exactly
microphone,
right.
right?
It
That's what
isn't that your system isn't working. It's just that I didn't have the lavalier on. So if I have the lavalier on and this is plugged in, if I have the lavalier on and this is plugged in, now you can hear me, right?
Yes, I can hear you, but isn't that the microphone on the computer and not the lavalier?
No, when I
switches to the lavalier.
yeah, it switches to the lavalier when I have that little widget.
It looks like your laptop does it automatically now, which is good.
Yeah.
So anyway.
But the problem is you can't hear through your computer when someone in the room is talking in the lavalier.
I need to figure out how to make the lavalier.
Okay, I just lost you again. No audio. No audio.
Okay, can you hear it
Leave it
now?
on.
Yes, now I can hear you.
Okay, because this is the microphone and I stuck it in the charging box,
Oh,
which wouldn't means it wouldn't work.
So let's figure this out now. So what we need to figure out is how to make the lavalier audio come over the.
yeah, Greg, maybe there's a sound setting that the lavalier can play through the computer speakers.
Computers. Yeah, sure. Let me try and figure it out.
Okay.
I know nothing
Greg's
about Apple
Greg's at the
computers.
wheel.
If this computer, I'm sure I can figure this out, but I know nothing about apples is my problem.
Okay, the
I don't
Apple
have to
menu up
navigate
in the
with this.
upper left-hand corner, you click on that and there'll be preferences or settings.
So I see audio. Here we go. So if I click the audio button, there's a microphone setting within Zoom. I can select a microphone, which is great. There's also a select a speaker.
Speaker, yeah, that's I think where you need to be.
So I see MacBook speakers or 6 Series 58. The question, what the heck is that?
Yeah, what the heck is that? Maybe that's the lavalier. Is it plugged in? Is it plugged into the USB now?
I wonder if it's the TV. That would be good if it was.
Could be.
Let me try selecting that. Now the question is.
How do we tell if it's
Yeah, well, would you hear me through are you hearing me through the computer or the TV?
Yes. That's the
Testing.
TV.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 9, 8, 7, 6, 5.
We can successfully use the TV speakers instead of the laptop speakers, which are way louder than your laptop. Now the question is, can we hear the lavalier audio over that? So let me go out of the room.
I'm hearing you loud and clear.
Because I'm holding a mic.
Yeah,
The question is, can Joanne hear me over the TV? That seems to be what's not happening, right? Joanne, can you hear me over the TV?
The mic is a lot clearer than the computer microphone.
So Joanne, you cannot hear me over the TV?
I can't hear you.
Right, you're not hearing me speaking on the TV, right? So that's the problem we need to solve.
Yeah.
And the thing is, this switch being in this position is supposed to make that happen.
Try the other position.
Yeah. How about now?
The volume turned up on the TV.
It did send up a message. Your default microphone has changed.
And Greg is coming through loud and clear. Joanne, you are not.
Yeah.
Oh, I'm not.
Well, because she
Now
now.
you were.
I don't have a mic. I have to have a mic.
There's a reason we have 4 of them.
Now, what about now?
Yes, I can hear you now. That's loud and clear.
Okay.
We weren't hearing it through the TV either.
No, before because we just figured out how to do that.
Okay.
Now, the real problem is that next week we have Alex doing the program, and we have the complexity of the issue with his laptop. But supposedly this is going to bypass.
Yeah, he's not going to have a problem.
His laptop issue. Okay.
And now we know.
Will that microphone, is there a clip where you can wear it on your?
Yeah.
Clip it to your shirt?
Yeah, I had it on my key.
So there are no audio problems with this system for the Zoom people at all.
Right.
The only thing we're trying to do is we're trying to figure out how we can broadcast the mic audio into the room, which we should be able to do. And I'm pretty sure that it works on my laptop, but we don't know how to make it work on an Apple laptop as yet. So if we can switch the meeting host to my laptop temporarily, I can prove that.
You want to try?
Because I think it works just fine over my laptop, and then we might have to just figure
you
out.
have to log on.
Yeah, I'll log on,
Yeah,
and then
okay.
I'll take over as meeting host, and then we can test the theory, and then we'll have to figure out why it doesn't do it with an Apple.
Greg's going
afterwards,
to take over as meeting host.
Yeah,
Always scary.
I want to work all this out now because I keep coming in every Sunday thinking we're going to resolve this.
I get it. I have no place I have to be.
We need to figure this out
Ed
once
is
and
being
for all.
flexible. Alice standing by. We're right here with you, Greg.
I'm out here in Utah with a perfect signal. Wi-Fi is working real good out here.
So
the weather? I don't have a mic, so I have to shout. How's the weather out there?
you can take your own mic out of the thing if you want. We got 4 of them.
He's not really there. You know that, right? Okay.
How's the weather where you are physically?
Where I am physically, I'm in Delray Beach, and it's mostly cloudy and somewhere in the 70s. It's not raining right now, but there's supposedly a chance later this afternoon.
It's Florida.
Yeah, exactly. Yeah. Don't like the weather? Wait a bit.
I was worried about the weather coming down yesterday because I thought it was going to be setting up in the rain. And I ran late and missed the rain. Just got finished setting up and it arrived again. It's Florida. You don't like the weather? Wait 5 minutes or move a mile.
Now you came down in your RV?
Yeah.
Okay. How
Well,
big is it?
You're just a rambling guy, eh? You just
36 foot. Looks like a bus. It's actually
You have a
Ford.
tow, Al?
Yes, I do. I tow a plug-in hybrid.
Oh.
The only one you could tow, the Ford Escape and its predecessor.
Will the RV charge your EV?
Yeah, but that's like using any generator to charge the EV. It's a waste. It doesn't charge it when you're rolling down the road with regenerative braking. But I do have a charger built into the motorhome. I ran a 220 line to one side of the motorhome. So when I'm plugged in at the park, I can plug my car in and do level 2 charging, which gives me a full charge in about 3.5 hours.
Very good.
I have to move at the outlet because I can't park close enough where I am now. The last park I was in, it was free. This one, I'll be paying for the electricity.
right.
At home in Ohio, it takes, if I charge overnight, 83 cents worth of electricity to give me 37 miles. That's pretty good mileage.
Oh, are we still recording? We can stop recording them. You know?
Oh, golly, we sure are.